Uh, Carl was a fellow at the Naval Medical Center when I was at NCI, and we very early heard about his talent, and it became clear that that was not a mistake. He's done some of the remarkable work on T-cell vaccines, and we're going to hear an update today. Thank you.
Well, I'm going to stay with the theme of targeted therapies, but this will now be an immune-based approach. And what we're working at is really, I think, an ultimately personalized therapy, which is one deriving from the patient's own immune system. You will hear two approaches between Suzanne's talk and mine on how to enhance the function of the immune system. I think this addresses the reputational issue that oncology's had and rightfully deserve, that cancer vaccines don't work in the absence of, unless they're a virally induced tumor. So for tumors that are non-virologic, the immune system really in general fails with standard vaccines, and so the field's moving towards approaches that use synthetic biology approaches to enhance the function of the immune system beyond what it does in nature. So there are two main approaches now in the clinic in various academic centers to test engineered T cells for therapies of cancer. One involves transfer of T cell receptors. So this would be a cartoon where this is the T cell, and this would be the tumor cell out here. So the T cell receptor naturally can recognize, if it has an appropriate specificity, recognize tumors via the HLA molecules and peptides that are bound in the cleft of the HLA molecule. So this is the specificity of the T cell receptor conferred by these rearranged alpha and beta chains. And the natural immune system has very low affinity for self-tumors, the mutations that occur, but it's possible to enhance the affinity of those T cell receptors, and trials are underway to test this by both at academic centers and several biotechnology companies. The main limitation of this is then that you create a bispecific T cell, that you have the natural T cell receptor and then the introduced one, is that T cell receptor has to match the HLA of the patient that you are going to treat, so that both the tumor has to express the antigen and then the HLA has to be matched, similar to the immunogenetic issues that we have in bone marrow transplantation. The other approach being used now is so-called chimeric antigen receptors, or CARs, as I'll refer to them. And in this instance, then, an antibody is introduced into the T cell, and with signaling domains downstream. And I'll describe more of this. The idea then is that the antibody then binds, and it binds with higher affinity than does a natural T cell receptor, and it can then bind now to the surface proteome or glycome of a T cell, of a tumor cell. So the advantages of this approach are that it's more off the shelf. You don't have to do HLA matching. You still need to know what the targets are on the tumor cell that's being attacked. And in addition, then it becomes MHC independent, because antibodies they recognize natural structures, native structures, and not MHC peptide complexes, unlike here. So CARs were first conceived by Zelig Eshar and colleagues at the Weizmann Institute back in the late 1980s. And I think it's a really good example of issues in translation, because it took about 20 years until human trials were conducted. And that was because it required the development of efficient T cell culture systems and gene transfer systems in order to create these modified T cells. The first trials that were reported in cancer were reported in 2006, one done at the NCI by Michael Kershaw and colleagues targeting folate receptor 1 alpha, and then another targeting done in the Netherlands targeting carboxy anhydrase 9 in renal cell carcinoma. Both these trials showed safety in a phase 1 setting, but they had no clinical efficacy. And we now can recognize why that was, which was that the T cells that were transferred to the patients didn't engraft and proliferate in the patients. They had half-lives on the order of a week or less. So what we and others did then was to then work on, making more second-generation chimeric antigen receptors to improve on those initial results. And when I was at the Navy Medical Oncology branch, we discovered that CD28 is actually the gatekeeper for T cell proliferation, and we then were able to develop a T cell culture system based on that using CD28-directed antibodies. And we conjugated polystyrene paramagnetic beads that are cell size or 4.5 microns. We conjugated antibodies to anti-CD28, which binds to T cells, and to the T cell receptor, which is using the anti-CD3 molecules to produce then an efficient T cell culturing system. The T cells are grown for 10 days in vitro in our current manufacturing approach, and then removed by passage over a magnetic field. And you can see this iron-colored. These are the beads. It's a 6-log 10 removal of the beads by passaging over this magnetic field. We first treated our first CAR patients in 1998. They initially had HIV, and I'll describe briefly these results, and then began trials in cancer patients in 2010. And the product that we infuse is infusion ready. It's cryopreserved in DMSO, similar to a stem cell transplant. So the cells are just delivered to the patient's bedside, thawed in a 37-degree water bath, and then infused over a 15-minute period, similar to a blood transfusion. So that's the basic drug. It's patient-specific. We have done a few identical twins where syngeneic donors are used, but in general, they're autologous. So the first CAR trials were done in patients with HIV using a CAR called CD4z, and this was an attempt to redirect T cells to be specific for HIV, because CD4 is a high-affinity receptor for the envelope protein of HIV. So 3 trials were conducted with a culture system that we developed and reported, and starting back in 1998, in patients that had HIV, testing either a single infusion of these CAR cells that were retargeted for HIV, or 3 infusions, or a single infusion with IL-2, so various dosing and scheduling. And then we recently have analyzed the results of, and long-term persistence of the CARs in these trials, and found, much to our surprise, that these CARs persisted now out past a decade in the patients. And in 41 out of 43 patients that we analyzed on the trial, they're persisting out to at least 11 years at high levels, at about 0.5% of T cells. So these are higher than memory levels are normally of T cells after a vaccine. The survival of these T cells actually exceeds then that of natural T cells that you elicit from a vaccine. When you do linear effects mixed modeling, finding the CD4z CAR using PCR approaches, it's quantitative, the survival half-life is more than 16 years in these patients. It ranges from 17 years to 24 years in the 3 trials. These were published earlier this year in Science Translational Medicine. So these T cells are predicted to last at this decay rate, which is very near 0 to the order of a patient's lifetime. So similar to a natural vaccine, if you have an effective vaccine, you can then protect people for a lifetime. We found now these CARs can persist, projected on the modeling, for a lifetime in HIV patients. And most importantly, there have been no severe adverse events. So there have been issues of would there be genotoxicity once you use integrating retroviruses or HIV to introduce T cell genes into T cells, and there have been no SAEs in more than 540 years of patient follow-up. So this platform is safe, at least in HIV, and using a CAR that's a so-called first-generation CAR. And that would be shown here, a so-called classic CAR. This is with a single signaling domain, and that was what was first used in the initial cancer trials that I mentioned, that mimics the T cell receptor. It's a zeta chain. It activates the RAS signaling pathway in T cells. What we and others now in the field are testing are more souped-up CARs, either sports CARs or muscle CARs, which, if you would say, would, in this case, 2 signaling domains and now even 3 signaling domains or more. As I'll show you, I think in leukemia that we don't need more souped-up T cells than what we get with this. But for other solid tumors, such as lung cancer and pancreatic cancer, we and others are working on even more potent T cells that have more enhanced proliferation and resistance to the tumor microenvironment. That's the goal of this T cell engineering. So the initial target for testing engineered T cells, both the FDA and the field has concluded, should be in B-cell malignancies. And that's because we have a dream target, which is, in this case, CD19 or CD20, which, rituximab targets this. CD19 is on all mature B cells, but it's not on the hematopoietic stem cells. So that if the CAR T cells are effective, we expect B-cell aplasia. And in fact, that's one readout of mechanism of action is B-cell aplasia. But we don't expect to have aplastic anemia as a side effect. So this is a good proving ground for new T-cell designs. And there are about 7 trials underway at various academic centers now testing these CAR T cells in B-cell malignancies such as this, using CD19 as a target. It's also possible to test other molecules as targets that have a more restricted lineage differentiation, such as CD22, which is on the more mature B-cell malignancies. So the CARs that we made in my group are shown here in cartoon form. They have an antibody, a single-chain variable fragment that binds with high affinity to CD19, and then it's expressed we tested a number of signaling domains, CD28, 4-1BB. 4-1BB is also called CD137, and it's a member of the tumor necrosis family members, so that it activates signaling through TRAFs in T cells when it naturally is bound by its ligand. And we tested them in all combinations pairwise and then all 3 here, and expressed them brightly on the surface of primary T cells shown here using an HIV-based lentivirus. And what Michael Malone in my lab found was a surprising result, which when T cells are activated with the beads that I mentioned, they undergo, this is population doubling, so this is an exponential, semi-exponential plot. They begin doubling, and with all CARs or with mock transduced T cells, they then rest down unless they're restimulated or IL-2 is added. But one of the CARs continue to grow for about 20 days, and that's the one that we took into the clinic. It has 4-1BB and T cell receptor zeta, and that's the one we're using now in leukemia patients. The initial patients treated had CLL. This is the survival experience at MD Anderson in a slide from Michael Keating. If you look at garden variety CLL from presentation, it has a long survival. It's generally not curable. The treatments now, the one curable therapy has been an allogeneic transplant, but it has high morbidity in patients with CLL because they're generally elderly with about a 50% incidence of chronic GVH. And if you take patients who've, second line who failed fludarabine, the survival then becomes more like that of pancreatic cancer with very poor prognosis. You can also identify now patients with CLL who will have poor prognosis based on genotyping of the tumors, such as p53 mutations, which are 17p deletions, splicing factor B3, and ZAP70 kinase activation, all predict poor prognosis in CLL, as well as TCL1 expression. So this is the trial that we have ongoing, and I'll show you where we are on the status of that. We use an HIV-based lentivirus. It's the first time HIV has been used as a vector in cancer patients in this trial. We've used it previously in HIV patients. And so here we use the HIV to introduce the CAR. Then we have the 10-day manufacturing process that I mentioned with the autologous T cells. So the patients are apheresed. And then the beads add this lentiviral vector, and then 10 days later the cells are frozen, and after they pass release criteria, standard FDA mandated release criteria such as sterility and endotoxin, they're ready for infusion. To be on this protocol, patients had to have failed at least 2 different FDA-approved regimens for CLL and have progressive relapsed refractory disease. The patients were allowed on this protocol dealer's choice chemotherapy given in the week before, and I'll mention what that was and has been in our patients. And then they're given the T cells split out over 3 days currently. We've done that as a split dose infusion because we didn't know if the T cells would cause cytokine storms in patients when we infuse them into patients who have circulating B-cell leukemia. So they're going to see, unlike natural T cells, they'll see antigen when they enter the circulation if patients have circulating leukemic cells. And so we have given the cells initially at 10% on the first day, and then 30% of the dose the second day, and 60% the third day, in order to test whether that would occur. And we reported the results from the first 3 patients last August. At that point, we had run out of vector, We now have, as I'll show, treated 12 subjects that I can report on to you. So the study is ongoing. We'll finish when 14 patients have been enrolled. As of now, 12 have been treated, 10 with CLL and 2 with ALL. The first patient was treated in July of 2010, and we have 7 CRs, 2 PRs, and 3 non-responses to date. And importantly, none of the patients have relapsed, which has actually been a big surprise now. We have out past 2 years initial subjects who are treated. And given that it's a monotherapy against CD19, we expected CD19 relapses to occur, and to date we have not observed that. And we treated over 100 mice now with humanized mice where we've given infusions of ALL from patients and then treated with CAR T cells. And we've only had in 1 mouse relapse of CD19 negative tumors. So it looks that B-cell malignancies have a very big need to retain CD19 expression, which to me was a surprise. We thought we were going to have to use combination CARs directed against a variety of molecules. In fact, we've made those to have in case we do get escape mutations. So these B-cell malignancies may be unique that way. We expect in solid tumors that, in fact, we probably will have to use combinational approaches along with probably targeted kinase inhibitors. So our issue now is, and this is actually Novartis' issue, we treated 12 patients. We're treating about 1 a week in our boutique setting at the University of Pennsylvania. We have a small manufacturing facility. And how do they outscale this now to treat thousands of patients? And what is being developed are robotic culture systems so that process that I described to you can be done it doesn't need scaling up, it needs what's called outscaling, which is just doing it a lot of times, what we do right now, growing 10 to the 8th T cells, the usual dose. So these are then the results from our first 3 patients that we reported last August. The patient 2 here was a 77-year-old gentleman who had p53 deficient CLL. His leukemic count is shown in red, or as his absolute lymphocyte count, with the upper limit of normal showing on that dotted line. And he was given 3 cycles of rituximab and bendamustine, and was refractory to that, as you can see. He had p53 deficient CLL, which is usually refractory to chemotherapy. When he had the CARs given, he then had a very rapid clearance of the CLL cells from the blood. And in fact, 14 days later, he also had 104-degree fever, an FUO, which at that time we didn't know the basis of it. He was treated with corticosteroids because he began to have angina. He had stents and had pretty significant comorbidity with atherosclerotic coronary vascular disease. He had a long-term partial response. We're not sure if the corticosteroids aborted a complete response, and we didn't have vector to retreat him. The other 2 patients on that protocol initially treated had complete responses, which are maintained until today. This is an example of patient 1 who had CD20 immunohistochemistry in the bone marrow here showing leukemia, and then at baseline, and then 6 months later, there's 2 interesting things. He has no leukemia, which is nice, but he also has no B cells. He has a very deep B cell aplasia. And in fact, all of our patients now who have had complete responses are on intravenous gamma globulin replacement prophylactically because they all have deep B cell aplasia and, in fact, are making no antibodies or low levels. So, and they all had clearance of lymph nodes, shown here in patient 3 who had bilateral axillary adenopathy. The responses are rapid. We don't see delayed responses as in vaccines. These are maximum within a month in general, more like a cytolytic response. So the summary of the initial reports, which were published here, were that all 3 patients with relapsed refractory CLL had and 2 of the 3 had p53 mutations, had very dramatic responses. There were 3.5 to 7 pounds of tumor per patient that was cleared. That could be assessed by quantitative assessment of the bone marrows, CT scans, and blood. And we could show for the first time that these CAR T cells were so-called serial killer cells, that each T cell could kill on average more than 1,000 tumor cells. And it does that either intermitotally, the T cell itself, or through its progeny. So the T cells divide when they see targets. And as I'll show you, the pharmacology of these cells is that they expand between 1,000 and 10,000 fold in patients. to reach an effector or target ratio to be able to eradicate tumor. And then, as I mentioned, the remissions have been durable to date. So it's early on, but it looks promising in B-cell CLL. So this is the pharmacology of these CARs in the first 3 patients, where the cells are infused here. And then this is on a log scale measuring the transgene 4-1BB and zeta that's in the genomic DNA of the T cells from the patients. So this is drawing blood from the patients. And quantitatively, we can assess down to 10 copies per microgram of genomic DNA. And what you can see is that they rapidly increased these CAR T cells in patients, reached a peak between 2 to 3 weeks after infusion, and then remain present in all patients out here to 6 months, and I'll show you now out past 2 years. So this is very different than the initial pharmacology reported with the first generation CARs, where the cells had half-lives of less than a week. Here they actually proliferate in the patients, come to a plateau, and then when tumor's gone, they then stay at memory levels. And we can also show that they continue to express antibodies. They have, as I mentioned, a single-chain variable fragment that binds to CD19. And this is staining with an anti-idiotype reagent on patient 3 here, shown at 2 months, and at 6 months, the surface of CD8 T cells. And here at 6 months out, 17% of this patient's T cells have the anti-idiotype, the anti-CD19 expressed. So from the pharmaceutical point of view, this really could be a game changer, because a single infusion of T cells can be given to deliver antibodies, and now they're, at least we know, for 2 years in patients. So you don't have to have the recursive addition like we do with current antibody therapies, or every 3 weeks to a month. A single infusion of T cells was able to maintain long-lasting antibody expression now in our leukemia patients. And we have an idea that, at least on these early results, that the proliferation of these CARs is related to their clinical responses. So this, again, is the pharmacology showing the copies of these CAR in the peripheral blood. And patients who have had CRs have more than 1,000 CAR T cells per microgram of DNA by 3 to 4 weeks after infusion. And non-responding patients here have not had that. So we believe that the T cells need to proliferate in the patients in order to mediate complete regression of the tumors, and that in some cases the T cells may not proliferate, perhaps because of senescence, telomere erosion, and this is an issue we're looking at in elderly patients, if this is why these patients haven't responded. As I mentioned, the CARs do proliferate, and this is detecting them in patients 1 and 3 now at 2 years after infusion. This is CD3 staining here. And they're actually more in the bone marrow than there are in the blood. So we've found in general, this patient, for instance, has 15% of his T cells in the marrow are CAR cells, and in the blood at the same time, it's 3%. So we believe that the bone marrow is a niche. We've found in all our patients that there's higher levels in the bone marrow than in the blood. And we've also found sustained B-cell aplasia. This is, at baseline, patient 1 staining for CD19 and CD20. So this is leukemia cells in the blood. Also, normal B cells would be in that gate as well on flow cytometer. And at 18 months, you can see there's nothing. He's got B-cell aplasia and also, fortunately, no leukemia as well. And so these initial studies have been informative. We've learned a lot about safety and feasibility, and I'll mention more of that. We expected the CAR cells to be rejected, and that's because we've used a mouse monoclonal antibody as our CD19. And so we expected the patients to make a HAMA, or human anti-mouse antibodies, against the CAR cells and reject them, which has happened in previous studies. And that hasn't happened. We believe that's because of the very deep B-cell aplasia these CAR cells are inducing, and the previous CAR studies have not done that, so that they can't reject their own cells because of they become tolerant to them because they have the CAR cells have made a niche by eradicating the B cells that would have rejected them. We're not sure of the mechanism of long-term persistence. It could be antigen dependent, in that case by CD19, so that as nascent stem cells become wannabe B cells in the bone marrow, it may be that the CAR cells live there in the bone marrow and basically pick them off as bears do with salmon sometimes swimming upstream. As they come by, they then prevent the B cells from emerging, and that may be a self-vaccination approach, is how the CARs are maintained, or it may be antigen independent. And So some of these cells that we are transducing and infusing to the patients are already memory cells. For instance, they may be a flu memory cell that then becomes a CAR cell as well. So they continue to express their natural TCR as well as a CAR. Those cells are already programmed to be long-term residents in patients. And so that's a cell intrinsic effect that's been done because they've been previously, for instance, become a memory cell. So we don't know this mechanism yet of persistence. We also don't know if there's a dose-response relationship between the tumor burden in patients and then the CAR proliferation. We suspect that the tumor actually drives T-cell proliferation, and then therefore the effective dose may vary with patients' tumor burden. At this point, we have not seen a strong dose-response relationship because unlike with standard drugs, because these cells can proliferate, what we found is that it's pretty much independent of the dose. We believe you have to have a threshold of cells that are infused, as with stem cells when, for instance, a transplant's done. And we don't know the long-term safety yet of the cells. That, per FDA regulations, their patients who are treated with integrating vectors need to be followed for 15 years. And so these patients are all going to be followed for that period of time to look for long-term safety. But by virtue of what we've seen with our CAR studies in HIV, where we began those a decade ago, we think that the safety, the basic platform, that the patients will have safety. The main issue probably is B-cell aplasia and the need for ongoing gamma globulin replacement. Finally, CLL, is an indolent disease until late stages. And so what would happen with CAR cells in a more rapidly growing malignancy? And we're now getting data in that in acute leukemia. And our first trial is what we call our PD-CARs. So we're testing now in kids with refractory and relapsed ALL, pre-B-cell ALL. And there are two subjects we've treated today at Children's Hospital in Philadelphia. So one, a 7-year-old with a high risk by virtue of karyotype who failed Children's Oncology group induction, then relapsed and had persistent disease after a number of salvage approaches. She was treated in April of this year at a point when she had 60% blasts in the marrow and then evidence of involvement systemically by scans. And so she was treated with 10 to the 7th CARs per kilo. And as I'll show, in this case, she got no further chemotherapy, and she now is in complete remission now, more than 6 months out. She had severe toxicity with a tumor lysis syndrome that I'll describe, beginning a week after treatment. So the second patient that we treated was given a dose reduction down to 10 to the 6 CARs per kilo. And this patient now also had a high-risk karyotype, but had relapsed after induction therapy, had an allogeneic transplant, of which there are no salvage therapies when you relapse from allo transplant. And her transplant was with a cord blood donor who was 4 out of 6 HLA matched. What we did in this patient was then treat the patient with the T cells that she had, which were actually allogeneic. So the T cells were derived from her cord blood donor, but re-derived out of the patient. This patient, interestingly, had relapsed after Blinatumomab therapy. Blinatumomab is a bispecific antibody that binds CD3 and CD19, is being developed by Amgen for ALL. And so she was refractory to 2 different courses of that. And we treated her much more recently in September, and she has also had a complete response, a very gratifying response. So the first patient shown here, this is absolute cells per microliter here of total white blood count, absolute lymphocyte count, and neutrophil count. So she was given split dose infusion here. The patient was aplastic. She'd had her previous chemotherapy 6 weeks before this. And what was gratifying is neutrophils came back 12 days after the CAR infusion, and these are all CAR cells. These are lymphocytes. Initially, they look like blasts, and they are blasts, but they're T cells, not leukemic cells, and as I'll show you. So this is bone marrow on day 6 after infusion. The T cells here, the CAR, there's a few dots here. These are actually specific. There's 3% on day 6 of CAR cells at a point when she still had in the bone marrow again. These are marrow T cells, so T cells that have already trafficked to the marrow. On day 6, she has CD20 and CD19 stain. These are blasts in the marrow. 23 days after the infusion of CARs, there's no one left in that gate. So there's a remission. It's actually a very deep remission. And the CARs, as you can see, have had an explosive proliferation. They divide, we believe, on about an 8-hour half-life, and they have now 75% of her T cells in her bone marrow are now CAR cells. So she has a very deep remission. By MRD staining, less than 0.01% of her cells are leukemic cells, and she maintains in a CR now, 6 months out. And this is at the time we treated her. She did not have known CNS leukemia, but I'll show you that the CARs have trafficked to the 2 patients we treated in the CNS, and then she's in first day of school here. She is now had as complications that I'll talk about of tumor lysis syndrome, which was severe and required support in the MICU from days 7 through 14. And she has had now a salmonella infection, which we believe is consequence to the B cell depletion, and that was treated with the antibiotics, and she's now getting B cell IVIG infusion. So the second subject, as I mentioned, was refractory to Blinatumomab and had a more primitive ALL. So this is a CD34 positive, DIM positive, CD19 positive tumor. She's given a dose reduction, had tumor lysis syndrome between days 7 and 13 after infusion, massive card expansion, as I showed. And interestingly, these are allogeneic T cells, and she had no GVHD, which was a potential risk. And a marrow then, and a month after, shows a complete response. She's now 2 months out and has normal counts. And interestingly, has this finding, as does our other patients. So this is trafficking the CAR here and following them by staining in blood, marrow mononuclear cells, and in CSF. So she had a routine tap at a month, and half the cells in her CSF, when you spin them down, are CAR cells. So they can traffic to the brain. We think this is very good news for potentially treating, for instance, glioblastomas. These CARs get to the CNS in 2 out of 2 patients now, where we've tapped, and they're higher in here in the marrow than they are in the blood, as we've seen in our adults with CLL. So we're having long-term persistence of these cells. This is day 87 now in the first patient with ALL. Here showing it CD3, that 30% at this point now are CARs. At day 7 to 14, 100% of her T cells essentially were CAR cells. Now they're being diluted out. We expect in kids, they will then have thymus comes back, unlike the adults. So the elderly adults are kind of like skid mice who have no B cells and very few T cells. And their thymus has not come back. So they're lymphopenic, they have lots of macrophages and natural killer cells, but very few T cells and no B cells. The children, we expect the thymus will come back. Usually the kinetics are 6 months or so to 12 months after chemotherapy. The thymus regenerates, and then new T cells come out. And then we expect that immune reconstitution then will be different than it will be in the adults, and that probably these CAR cells will be diluted out as new thymic T cells begin to emerge. So we have had severe on-target toxicity. It's delayed to the time of peak CAR T cell proliferation, and it's cytokine release syndrome. Manifests with high fevers, nausea, hypotension, and in the serum, high levels of interleukin-6, interferon gamma, and then TNF and IL-2 at lower levels. It's now been treated initially with steroids, but our current drug of choice is tocilizumab, which is anti-IL-6, and has a remarkable effect on stopping this, and I'll show an example of that. So IL-6 signaling neutralization stops this. We think if we give it too soon, though, that it may stop tumor rejection. And so this is how we're managing our patients currently, generally treating them between days 10 and 12 with tocilizumab. Now, we're hoping to get to a point in CLL, for instance, where the patients are treated with targeted inhibitors, such as a BTK inhibitor, and then not have high tumor burdens. We believe at that point then that the patients won't have the cytokine release syndrome. But right now, it's a lot of shake and bake. They have 103 to 105 degree fevers until the tumor goes away. And this was the first patient that we had experience with this in, this patient was given a small number of CAR T cells, which was 15 million. That's literally the size of a T75 flask. And he'd had previous CAM-PATH therapy, which is alemtuzumab. He had almost no T cells to start with, so we had a manufacturing failure. He got 15 million cells infused here. We thought nothing would happen, but then he was hospitalized 3 weeks later with delayed tumor lysis syndrome, and it was really incredible. It was not trivial tumor lysis syndrome. It was required rasburicase therapy. He had transient renal failure. It all resolved within a month, and he now remains a CR now 2 years out. But it was completely unexpected that we could see a delayed tumor lysis syndrome. It's not been experienced in medicine before. We've now had this happen to a second man who's 78 years old, 51 days after we infused the CAR T cells, and then accompanied then with an induction of a complete response. So where we are right now is, to try to complete this phase 1 study. That will happen later this month when the 14th patient will be infused. Then we're analyzing a number of issues in the products to identify biomarkers. We're then going to begin a dose-finding study because we've had CRs at this point with a 2-log range in the number of T cells we've infused. So we're not trying to identify a maximum tolerated dose, but rather an optimal biologic dose. So trial design is different for these kinds of cell-based therapies than it is for cytotoxics. And then we're going to begin larger phase 2 trials in both CLL and ALL that Novartis will conduct. So these, in summary, CAR T cells can persist and remain functional at high levels for at least a decade in HIV patients and at least 2 years in patients with leukemia. They have potent and durable activity at least for 2 years in CLL and now out to 6 months in childhood leukemia. They do induce what we expected, which is long-term B-cell aplasia. We don't know whether we can kill these CAR T cells off and then have the B cells come back. And that's an issue that we're going to resolve, whether or not it's necessary to have long-term surveillance. That's the strength of the immune system, is that it can have long-term surveillance and potentially how it can synergize with targeted therapies. And so we don't know. There may be other strategies that don't require long-term B-cell aplasia, but at this point, that's what we have. We've had on-target cytokine syndrome in patients, which is self-limited and may require cytokine therapy, such as anti-IL-6, and we think that engineered T cells are probably one of the first examples of successful synthetic biology where the function of cells can be enhanced over what they do in nature. A number of people have been involved in these studies at the University of Pennsylvania and at Children's Hospital in Philadelphia. So David Porter is the PI of the trial at Penn, and Stephen Grupp at Children's Hospital. Bruce Levine's done the cell manufacturing, Michael Kalos, the immune analysis, and a number of people have helped both with reagents and with patient care. So I'd like to thank you for your attention.
I've lost my microphone. I'm not sure I need one. There's going to be a lot of questions. So, I'll open it to the audience. Susan.
Maybe you could hear me from here. So you showed So Carl, you showed in your diagrams that the CARs are single-chain constructs, but do you know if they dimerize in vivo, and do you think that makes a difference in terms of potency?
Yes. So the question is about the molecular structure of these CARs, and they actually, we can show that, so we have various transmembrane domains. For instance, CD8 alpha is what we're using here. Sometimes other CARs use Ig hinges and transmembrane domains, and so they dimerize. We can show that by reducing, non-reducing blots. What we don't know is the initial signal transduction, and in fact, this is much higher affinity than a T cell receptor, which is micromolar. T cell receptor, barely you can show binding by BIA core measurements to its MHC HLA target, whereas here there's about 10,000 copies of the CAR on the surface of these T cells, and when it binds to CD19, it's like Velcro. I don't understand how the CAR ever releases, how the T cell ever gets away from its target to go because of this. When we've seen it under a microscope, it's an amazing, very high, it just looks like they're glued together. So there's a lot, we think a SMAC probably, with the T cell activation complex. The normal T cell receptor, focuses when it hits its target and it comes together in something called a SMAC, and then it excludes phosphatases and then it assembles a signaling and then releases perforin and it kills its target. Somehow this works as a surrogate to do pretty much the same thing.
So, Carl, I'm flabbergasted. Is there any limit to what this can do? I mean, is there any obvious tumor that won't potentially respond? All you need is an antigen, right?
So, first of all, we have in vitro data that, for instance, in lung cancer, the CARs, if you retarget so that they can bind to a lung cancer cell, because a T cell receptor in general won't, they are potent killers of non-small cell lung cancer. The issue then becomes, do the T cells traffic there? And then can they maintain activity? It's as you know, I mean, a lot more immunosuppressive in the environment of, say, lung cancer or pancreatic cancer than leukemia. So I think a baseline hypothesis now is that diseases that can be cured with an allogeneic transplant probably can be cured with autologous CAR T cells. So, it's dependent on the targeting construct. For solid tumors like this, it's early days. And
what about lymphomas, like
Sure.
B-cell lymphomas? It would seem to be.
So Steve Rosenberg's had responses in diffuse large B-cell lymphoma, and Ron Gress and their group at the NCI with CARs similar to what we're doing. And we're going to begin a trial for both mantle cell next year and diffuse large B-cell. So hematologic malignancies, I think, will be druggable, if you will, with CARs this way. Solid tumors, it may well take other adjuvant approaches to maintain the activity of these cells. And I think one very thing we're looking forward to is a therapy you're going to hear about from Suzanne to maintain activity.
Would you like to briefly expand on this issue of just trying to eliminate the B-cell aplasia? Are you thinking about ways of replacing the B cells afterward without CD19 or some way of getting
there's a
around?
couple of approaches on this B-cell aplasia. So one would be use of so-called conditional suicide constructs, and there are a number of those have been developed. One that Malcolm Brenner and David Spencer published in New England Journal last year, where they used allogeneic T cells. They put in, with a dimerizer actually initially made by Ariad, they could show that they could activate caspase-9 and then kill the CAR T cells. Or in this case, they used just allogeneic T cells. They didn't have CARs in there. So I think it will be possible to kill these off, and then B cells come back. I mean, that's been within, as you know, 3 to 6 months after, for instance, after rituximab, we know the kinetics in adults when B cells come back. So what we don't know in these cases with CLL, for instance, is it tumor come back? Is it a sterilizing effect, or is there dormancy, of which, unfortunately, there's good data for tumor dormancy in humans, I think, now. So there may be it may be possible to eradicate the CAR T cells. It may be, then there's replacement therapy of gamma globulins. Malcolm Brenner has a clever strategy, which is target, for instance, as you know, these leukemias are either lambda or kappa positive. So you could just target the lambda or kappa with a CAR and then leave basically half of your repertoire there. So there's going to be, finer tooth approaches that will be developed, I think, to make it so that we don't have B-cell aplasia.
Long-standing toxicity. Marty.
Carl, that was beautiful. I mean,
thanks.
You saw Bruce and I gasping during your presentation. This morning earlier, we heard some beautiful presentations on precise medicines, in particular the crizotinib story as an example. And if you were to use that precise medicine to debulk the tumor, I'm sure you're thinking of then following on with immunotherapy. That could also perhaps be very exciting in melanoma as well. Would you care to speculate on that? And Alice to my left may also want to rejoin her.
I think that's where we certainly want to go, because we think then the side effects are less. The potency of the immune system is for long term. So I think there's tumor induction, and then consolidation approaches that you can envision. And so in the case of CLL, I mean, the landscape, I think, is going to completely change. There will be, I think, a chemotherapy-free approach, if everything works, to CLL, where the patients never have to get fludarabine. It'll be something such as a BTK inhibitor, an mTOR, AKT inhibitor, and then T cells to clean up.
And then to avoid that tumor lysis syndrome.
Yes, so that's exactly what we want to do. The problem is right now is we can't get those patients. You know, we replied just like everyone else has been, you know, by FDA standard drug development. We got to start with phase 1 patients right now, but that's where we want to go. And I think that's right now the therapy we're using, and I mean, can only be done at a high-end medical center such as Mass General or our place, because they're as sick for a month. They're as sick as an allotransplant patient with this tumor lysis stuff. So I'm not downplaying that at all. You need to be ready to intubate them, you know, give them pressors, and then know when to pull the plug, like, for instance, this immunosuppression. So that we need to get to a point where we can treat patients with lower tumor burden.
So just one final issue of scale-up. You mentioned Novartis being involved. Do you see this as something that can be done on a much larger scale nationally?
So it's the actual process can be, so you cannot do a process that requires PhDs to look at every product. I mean, there's not enough PhDs in the country to do lung cancer. So the conclusion is this needs to be made robotic. And there are companies now working on, we use what's called a wave bioreactor right now, which is a gas perfused. It has real-time analysis of things like glutamate, pH, you know, and all that. And so, and then it can feed itself automatically. So I think that the culture process needs to be made robotic without, you know, so that it's becomes like a blood transfusion, and then that will then, I think, make it scalable just like any other drug manufacturer.
Any idea of the cost?
So the cost of goods, so it's interesting. So the major cost here is gamma globulin.
Really?
Yeah. So the T cells themselves, with
cheap.
us, you know, for our in-house cost of an apheresis and so on, it's $15,000 to manufacture the T cells.
That's a single dose of Rituxan.
Yeah, so it's about the same as 6 weeks of an antibody supply. So the actual cost of goods, that's not making profit, as you know, but the cost of goods is not a problem when you look at other therapies that are out there, because it can be a durable effect.